Okay, welcome everybody. It is again my pleasure as the interim chair of the Department of Physiology here at New York Medical College to welcome you all to the 9th Drs.Gabor and Harriette Kaley Endowed Lectureship. You know, we're delighted this year especially to have our speaker here in person. So it's something that we all have to get used to again. I mean, actually shaking hands and maybe even hugging people that we haven't seen for years because of the pandemic. So we're back, but we're of course in a hybrid format as well. And we have some of our most esteemed colleagues visiting us via Zoom. So speak slowly so that they can hear you if you have any questions. So as I said, we're delighted to have in person Dr. Csiszar from the University of Oklahoma College of Medicine. In just a moment, she'll be introduced properly. But before that, I want to say that this lectureship was established through the generosity of Dr. Harriette Kaley and her son, David Kaley, both of whom, at least Harriette is here today, and David may be able to make it. I'm hoping he can visit us by Zoom. You know, and they established this award to honor the life and legacy of Dr. Gabor Kaley, who many of you know was the chair of physiology at New York Medical College for almost 40 years, from 1970 to 2007. As one of those fortunate enough to have known Gabe, I can tell you that he was a remarkable scientist, a terrific leader, and an overall mensch. And he provided much great advice to his junior faculty and to his junior scientists in his laboratory. And, you know, today's speaker is in fact one of the many associates of Gabe over his career who have gone on to a spectacular career after working with him. And so we're very excited to hear about what Anna has been working on and all of her successes that she'll tell us about today. Now, this year's lecture is marked by two additional special items that I wanted to tell you about. First, as some of you know, NYMC has been putting up memorial posters around the campus to recognize some of the major contributors to the history of doing science and research at New York Medical College. And today we have a new one that has just appeared, which honors the life and work of Dr. Gabor Kaley. And so I encourage you later today to visit the poster, which is hanging in the Department of Physiology conference room. In addition to that, we also have a print from Andreas Vesalius that has hung in Gabe's office for many, many years without a lot of understanding of what it really was. It turns out this is a remarkable print made from the original wood blocks from the book on the fabric of life that Vesalius published in, I believe it was 1545. And these wood blocks were lost for many years and discovered in the 1930s again later. And the New York Academy of Medicine reprinted the books. And fortunately, because the wood blocks were ultimately destroyed in the bombing of Munich in World War II. And so this is one of those prints that came from the reprinting of the book from the 1930s. And it's also been beautifully framed and hanging in the physiology conference room. And I encourage you to go take a look. And there's a lovely little description of it that's associated with that. So we're grateful to the generosity of Harriette Kaley for making this gift to the institution. And I also want to thank Mr. Nicholas Webb, who's our archivist in the library here, who's done a great job in assembling both the poster and the print and sleuthing the history of the print, which was largely unknown to the other members of our department. So please come and take a look at that. It's a little gruesome. I was going to show you the slide, but I thought it was shortly after breakfast and maybe not such a good idea. But Vesalius really had a great message that was passed on to generations following the production of this book, and that's to study life and not study books. The prevailing understanding of human anatomy came from Galen's writings from hundreds of years before that. He was the physician in Rome, and it turns out that Vesalius discovered that that Galen's writings were based on the anatomy of non-human species, and there were a lot of things that are different. And so the virtue of Vesalius's work was to encourage people to really study the life around you instead of just learning things from books. Okay, so with that, I have the pleasure of telling you what the program is like today. So first, we're going to have a few words from Dr. Irv Zucker, who is the Theodore Hubbard Professor of Cardiovascular Research in the Department of Cellular and Integrative Physiology at Nebraska Medical Center. Then Dr. Harriette Kaley will say a few words about this lecture and its meaning. And then we'll have Akos Koller, who will remotely introduce our speaker, Dr. Anna Csiszar. And following Dr. Csiszar's talk, we'll have a session moderated by Dr. Elizabeth Barry, who just successfully defended her thesis, and we're already putting her to work. We will then have a presentation of the award by Dr. Mike Wolin, who's the head of the selection committee. And after that, we'll have some closing remarks from Dr. Messina. So in a final note, I just want to thank the efforts of the selection and organizing committee and the members of the physiology office who have helped make this a successful event today. So, Dr. Zucker.
Let's see. I hope you can see my screen. Let's see. Everyone see that okay and hear me? Can you hear me?
Hear you just fine.
Yeah, you're good. We can hear you.
Yes, we can hear you.
Okay.
We can hear you, Herb.
Thank you.
Hello. Yeah.
So thank you, Chris, for that introduction and for the privilege of allowing me to say a few words about my mentor, friend, and colleague, Gabor Kaley. As some of you may know, I was Gabe's first graduate student, and it really is an honor to participate in this lecture series. As I mentioned, Gabe was a mentor, influenced my career and those of many others. And I just wanted to highlight something that I think was within Gabe's philosophy and way of thinking. This is a quote by Steven Spielberg, the famous director, who said, "The delicate balance of mentoring someone is not creating them in your own image, but giving them the opportunity to create themselves." And I think this is a good quote on mentorship, and it's something that Gabe, I think his thought process was always front and center in terms of the people he trained. And over the years, I became a good colleague as I left New York Medical College and moved to Nebraska. We continued to discuss science on many occasions, both on my visits back to New York and at conferences. And then finally, Gabe and Harriette became lifelong friends, and we met up whenever we could at international meetings, and I'm so grateful for that opportunity. After I moved to Nebraska, Gabe visited us. He received the McIntyre Award, which is a yearly award we give out, and a lecture in 2003. And one of the things that Gabe said at that award ceremony is shown here. He said, "There are satisfactions derived through a long career in science that far outweigh what I imagine I would have accomplished as an MD. The pressures now in science are greater than ever," and of course that hasn't changed today. "Remaining creative is a full-time job and I relish it. As my biochemistry teacher, Albert St. Georgi, who won the Nobel Prize in Physiology or Medicine, said, a good scientist has to see what everybody sees and think what nobody else has thought." And that quote has stuck with me for many, many years now, and I'm thankful for Gabe to pointing that out. So Gabe's career really started way before me, but what I did in the lab was related to sodium regulation and the central sensing of sodium, along with Norman Levine and Gabe. And this shows our first paper in AJP before it was sectionalized. Gabe went on to collaborate with Dong Wang and Akosh and An Wang to study nitric oxide and the regulation of microvascular tone in skeletal muscle. And he's credited, the lab is credited largely with defining flow-mediated dilation, which is now a mechanism to evaluate endothelial function even in humans. And then went on to look at other specific mediators of the mechanotransduction process in the microcirculation. These other photos that you see here are Gabe and I on the lower left, Gabe and Harriette, my wife, and the Weinsteins at one of the physiology chairs meetings many years ago, and then at an EB meeting on the upper right with one of my postdoctoral fellows, my administrative assistant at the time, Cindy Norton, and Gabe and myself in San Francisco. And these are experiences and times that I will never forget. I just want to mention that Gabe, most of you know that Gabe came from Hungary, and one of his friends wrote a book about his experiences during the Nazi occupation in Hungary, when Gabe and his friends were rounded up and sent to a forced labor camp in Yugoslavia. And there was one quote from this book, The Reluctant Adventurer, that I would just want to highlight here because it typifies how I remember Gabe. Weeks passed and we began to organize ourselves. Sometimes we started daydreaming out loud. Kasia, Kaley, would talk of future feasts. He would recite the menu. Whoops, sorry. Whoops. Got ahead of myself. Let me see if I can move backwards. He would recite the menu out loud. A whole duck just for himself, garnished with oranges and stuffed with chestnuts, coated with chocolate, decorated with whipped cream, and basted with cognac. Every day he would add something to the recipe. And this is part of my experience knowing Gabe as a person. He endured that time and emigrated to the United States. I was very grateful, along with Akosh and Tom Hinsey, to be present when Gabe was awarded an honorary degree at Semmelweis University in Hungary some years back. And finally, I just want to say we had a little competition. Gabe and I traveled to China 1 year after I went 1 year after he did, and he came back and told me how high he went on the Great Wall, and I went a little bit higher, so I had to twist the knife a little bit and send him this picture that says Zucker 100, Kaley 99. So I thank you for inviting me to say these words, and I'm looking forward to the rest of the program. Thank you very much.
You look nice and relaxed. That's good. Now join you in the world of as Chairman Emeritus.
Thanks, Gabe.
Well, let's see. I'm Harriette Kaley, and I'm very gratified that we're here for the 9th annual Gabor Kaley Endowed Lecture. So let me tell you something about Gabor Kaley. My dear husband was Gabe to everyone who knew him, which sounds as though he was kind of casual about things like that. But Gabe was not truly a casual kind of person. Everything he did here in this department, in the medical school, in the process of science, he did with an intensity of commitment, devotion, even ardor. He did all that with his characteristic ebullient high spirits. How many people here knew Gabe? I know. Okay, well then I think you'll recognize him from this. High spirits, a great sense of fun, and an enormous capacity for friendship and humor and love. He'd lived through and experienced some of the worst of the Holocaust in Hungary, where he was born and raised in Budapest. And he said sometimes that it marked him, but it didn't scar him. You know, I'm a psychologist and psychoanalyst, so I sort of know better than that. Trauma like that scars people, but it did not destroy Gabe and his liveliness. One of his faculty told me once that Gabe's personal warmth, the creative unity that he'd created in the department, were why the physiology faculty regularly all ate lunch together in the departmental library. He'd created an atmosphere that was not only tremendously productive, but had steely bonds of friendship. One measure of what he accomplished was that he was the longest sitting chairman of any medical school department in the United States. He knew that that couldn't have happened without a really supportive faculty, and that gave him great satisfaction. Incidentally, as of today, there's some controversy over whether he was the longest sitting chairman of any medical school department or whether it was just a physiology department. And I maintain that it was of any medical school department. We'll slug that one out a little later. The measure of how deeply he cared about this department, about his work, and about science was that it was his fervent wish that we establish an ongoing process in his name that would continue his mission of teaching, informing, educating. Our son David and I were gratified that together with the leaders of this college and the department, we were able to fulfill Gabe's wish in the form of these lectures. And we're enormously proud and happy that it seems to be a going concern. We like to think of it as stretching far into the future. We joke to ourselves that one of the few good things about COVID was that it forced us to hold this lecture virtually or in a hybrid form, thus saving on costs while permitting everyone interested to attend no matter where they are. Another thing to joke about, in the beginning of these lectures, I noted at one of these presentations that it looked as though scientists were always men. In recent years, we fixed that. So it's very satisfying to have today's speaker here and a Hungarian woman at that. We're very tickled about that. So I want to thank the planners of the program and welcome our speaker and all of you present virtually for being here with this event that is so dear to our hearts. Thank you.
Dr. Koller now will introduce the speaker.
Thank you very much. I would like to welcome everybody. I'm from Denmark at the moment at the European Society of Microcirculation meeting. Therefore, I cannot attend in person. But it is a great pleasure for me to introduce Anna and talk about the history of Anna, or rather, I would say her story. And Anna finished his medical school in Budapest at Semmelweis University at 1998. Then after a little hesitation, she joined me and I became her mentor as a PhD student. I will tell you about that a little bit later. And after she finished PhD studies, she joined the New York Medical College and became the student of Dr. Kaley. He spent with him several years. Then she became independent. And then in 2009, she moved to the University of Oklahoma and then became an independent scientist and became a full professor there, an end-of chair of Donald W. Reynolds. And during this now 20 years, she published more than 200 papers and all very, very important papers. And she really brought in molecular biology into microcirculation. And also, I think she was one of the first who realized that inflammation is very important in microcirculation. And she has had several NIH grants. I counted around 10 and contributed to another 20. And on her own, she had many, many students, close to 20 students who was under her mentorship. And I think that you can look up her papers on the PubMed and then you know what she accomplished. But what I would like to really share with you her ethical standard, very high ethical standard. The reason she joined me and she picked me as a PhD mentor because she was working for someone else. But then she had trouble to repeat the experiment of that person. He was a very well-established scientist, but she couldn't repeat it. So she decided not to pursue anymore the PhD student with that person. Then she looked for me, find me, and then I was happy to be selected by her. And so she has a very high ethical standard in science. And I think that helped her to achieve whatever she achieved so far. And this is just the beginning. So I wish the best for Anna, and I'm very happy that I could share this with you. Thank you.
Thank you, Ákos, for the very nice introduction. I have to tell you that this is a very emotional moment for me. I'm back here on campus after 13 years, and hopefully I can show you that I tried to combine the legacy of the two basic and main research fields of the Department of Physiology. One was vascular physiology, the other neuroscience. So today, it's really a combination of the two that I would like to talk about. It's age-related vascular cognitive impairment, role of neurovascular senescence, and my journey in science that started here at the New York Medical College. Okay, so as Irv mentioned before, he was one of the first graduate students. I was one of the last postdoctoral fellow in Dr. Gabe Kaley's laboratory. And as I mentioned, The Reluctant Adventurer, the book by Thomas Reiter, that I actually own 5 copies because of Dr. Kaley. I was the kind of the reluctant microvascular physiologist. As Ákos said, I was originally an immunology student after finishing medical school in Hungary, with also a coveted residency position in the internal medicine department. But I wanted to do research, and I wanted to do it really badly. And Ákos helped me out as a graduate mentor and brought me here as a postdoctoral fellow. I joined the department in 2000, June, and with my immunology background, my pedigree wasn't really up to the standards of a very famous microvascular physiology department. But Dr. Kaley believed in me. He accepted me as a postdoctoral fellow, and he gave me freedom to do scientifically whatever I wanted to do. He believed in me so much that he sent me to the physiology course at the Marine Biology Center in Woods Hole in 2003, which was also established by Albert St. Georgi, as who was mentioned before by Irv. And after the first result, I got a topic from Dr. Kaley to investigate vascular changes or microvascular changes in aging. And that was in early 2000. And aging, it was just another topic, a very nebulous topic at that time. Nobody really realized what aging is and what is the undergoing biology of aging. It was like equal to vascular changes in hypertension or vascular changes in diabetes, just another topic. However, when I was in Woods Hole, I realized that this is a separate field of science. It's called the biology of aging. And Dr. Kaley actually supported me to go back in 2004 and participate as a student in the biology of aging course. Then from 2006 to 2011, I became instructor in this biology of aging course. And here my love of aging and vascular physiology were combined and launched a very successful career thanks to him. So when I first attended the biology of aging course, I didn't know much about the field. It was really a bunch of very colorful scientist characters who were just interested why we are aging. But by 2010, we realized that the Western world is aging, which means that the number of people who are 65 years or older is exponentially increasing. And if this trajectory is true by 2050, we're going to have more than 1.5 billion people who are older than 65 years. And as lifespan doesn't equal health span, our aging population is riddled with age-related diseases, chronic diseases, which are putting a huge burden on our healthcare system. And this was realized by NIH also, and a whole new field of science were born, which called geroscience, an intersection of the biology of aging and health span research, new ways to understand how common mechanisms, biological and cellular mechanisms governing aging might underlie the occurrence of pathology of diverse chronic diseases. And this geroscience field actually prompts the interaction between basic science researchers, preclinical researchers, clinical physician scientists, and public health advocates. And this is where I found my scientific niche as an MD PhD. So the importance of age-related cardiovascular pathologies can't be underestimated. In this recent review that my group published in Journal of American College of Cardiology, we tried to summarize the entire concept of geroscience. And the three roots are all of the different cellular mechanisms that are contributing to the biology of aging. Then eventually, as the tree grows, the trunk becomes different branches, which are the different age-related pathologies. And one of the major components is the vasculature, is the highway to all of the signaling mechanisms to different organs. Currently, the leading cause of death in the Western countries are still cardiovascular diseases, especially in the age group. This is still heart disease, which overpasses the mortality of cancer, stroke, and even diabetes, kidney disease, and even Alzheimer's disease has a very, very strong vascular component. So a couple of years ago, a good friend of ours, Philip Gorelick from Northwestern University, came up with this term vascular cognitive impairment. So what does it mean? Originally, it was supposed to encompass all of those cognitive impairment phenomena that don't fulfill the Alzheimer's criteria and has some kind of vascular pathology in the background, structural or functional changes that are contributing to neuronal dysfunction eventually, or neuronal damage. And the major risk factor for all of these vascular pathologies are aging. We have to be honest with ourselves that everybody of ourselves are going to be affected because current public health studies and epidemiological evidence shows that 95% of our seniors, community dwelling healthy seniors, actually show focal confluent hyperintensities, which are the MRI marks of vascular changes in the brain. So there is something going on. This is a traditional physiology department, and Dr. Messina always taught us that we have to understand the vascular role in every kind of organ function. And in the brain is particularly important. The functional and structural integrity of the whole brain depends on healthy cerebral vasculature. Why is it so? Because the brain doesn't have any energy or oxygen reserve capacity. Everything, all the neuronal function acutely has to be matched by acute cerebral blood flow changes and new supply of oxygen and energy. So this background figure that became one of our signature figure in my team shows a vascular cast of the brain. 600 kilometers of microvasculars are responsible to support the brain function. Literally every single neuron has its own supplying capillary. So my team in the last 10 years or 13 years since I left the medical college was very much involved identifying biomarkers or marking features of vascular aging in the brain. And we came up with the following phenotype, including both structural and functional changes that are characteristic, not only preclinical, but clinical aging as well. One of the first one is impaired neurovascular coupling. So what is neurovascular coupling? Neurovascular coupling is the functional hyperemia equivalent in the brain, which means that, as I mentioned before, the brain doesn't have neurons, doesn't have any energy reserve. Neurons are unable to store glucose. So their functional activity depends on the acute blood supply, oxygen, and energy supply. So what happens in the brain is upon neuronal activation, glutamate is released, which activates a calcium spike in astrocytes, which actually leads to the release of vasorelaxation molecules, and eventually NO, the major vasodilator that Dr. Kaley was so interested in. So in aging, because NO production is decreased and because free radical production is increased, NO is captured and scavenged, and the bioavailability of NO is decreased. This seems to be a little bit complicated figure, but the idea is very beautiful, simple, and logical. So as an MD, my research question was always relating back to how we can actually improve human health. And I always wanted to do a research that is meaningful for human health also, that I directly can translate. And a couple of years ago, I was fortunate enough to, beside of my basic science laboratory, launch a translational clinical laboratory that directly I was able to use all of the concepts that I investigated in research labs and all of the potential interventions that I investigated in my basic science research lab in a clinical setting. So this is a geroscience translational laboratory, which is directed by Dr. Andrei Yabluchensky, who sits here on this picture as one of the study subjects. And all of the parameters that I described as hallmarks of vascular aging or cerebral vascular aging, we measured in clinical settings as well. So one of the first parameters that we were interested in neurovascular coupling. So what happens with this neuronal activity evokes cerebral blood flow response? And in our translational laboratory, we showed that although neuronal activity, in this case, single finger tapping, can evoke cerebral blood flow increase, this is actually missing in or very much ablated in people who are 65 years old or older. The phenomenon was described, but we don't know the mechanisms. What's wrong? Obviously, in clinical studies, we can't really do mechanistic studies. So we needed preclinical models. And to measure neurovascular coupling, in mice, we used a paradigm called Whisker test or whisker stimulation, evoked cerebral blood flow change. What is it about is that this stimulating the whisker, which is the finger tapping equivalent in the human studies, evokes neuronal activation in the somatosensory cortex. And we were able to measure cerebral blood flow changes above the somatosensory cortex, as depicted here on this picture. We also showed that if we block this neurovascular coupling by blocking all of those pathways that I showed before, we are able to decrease the cerebral blood flow change upon neuronal activation. But further on, this has dire consequences, long-term consequences in terms of cognition. So by blocking cerebral blood flow change, neurovascular coupling, we actually induce cognitive impairment in our young mice models showed by Vaime's testing. How can we actually investigate this in an aged model? So we use the C57 Black 6 NIH-approved aged mice model, starting from 6 months to 32 months of age, which is approximately human equivalent of a 20 years old human going up to 70 or going up to 90 years. And we found that cognitive impairment in this mice model actually starts around 15 months old, between 15 and 20 months old mice, which is equivalent to 60 years of human lifespan. And this cognitive impairment actually plateaus there. In terms of spatial learning, which we measured with radial water maze, and the mice had to find an escape platform. And obviously at 15 months old mice were able to find the escape platform in a water-filled radial water maze. 20, 30 months old mice makes a lot of errors in terms of visiting the incorrect path. So we measured as a cognitive outcome measure or readout, we measured the errors, how many times the mice visiting the wrong arms, and how much time does it take for the mice to find the escape platform. And by this, we were able to characterize a learning curve and also memory function in our mice model. And we found both learning and memory is significantly impaired in this mice model of aging. At the same time, in parallel, we also assessed neurovascular coupling with the previously measured method. And what we showed that compared to the young mice, where there was a very nice evoked cerebral blood flow increase upon whisker stimulation, this increase was missing from aged animals. And also, this increase was completely NO dependent. If you block the NO signal, the increased blood flow change was completely ablated. We also showed that this reaction is also has to do something with oxidative stress. Oxidative stress, or the free radical theory of aging, one of the most prevalent theories of aging, which state that free radicals induce damage is actually the reason why we age. So we used the mice model where we had an antioxidant enzyme overexpressed in the mice called mitochondrial catalase. And by overexpressing this enzyme, we were able actually to protect in aging the cerebral blood flow response. So in conclusion, we can say that neurovascular coupling or evoked neuronal activity, evoked cerebral blood flow response is decreased both in human and clinical models in aging, and it's an NO-dependent pathway. The other phenotype, structural phenotype that we described was impairment of the blood-brain barrier. So what is the blood-brain barrier? Physiology 101, Dr. Messina's class. The blood-brain barrier is a structural barrier which is formed by endothelial cells. Endothelial cells are very special in the brain. They form tight junctions and prevent the brain from all of the noxious circulating factors. The blood-brain barrier is responsible to create an immunologically privileged position for the brain. It's formed by endothelial cells, but it's also formed by pericytes in the basal lamina and astrocytic end feet. So by investigating the blood-brain barrier, one of the earlier idea how aging impacts the brain came from Berislav Zlokovich's laboratory, showing that probably the blood-brain barrier disruption is actually precedes all of the neuronal damage and all of the neuronal dysfunction that we associate with cognitive impairment and dementia. His laboratory and his clinical studies showed also that in aging population, human population, there is an age-dependent increase of the blood-brain barrier disruption and extravasation of normal circulating proteins. One of them is thrombin or fibrin, which are part of the normal coagulation pathways. And he showed a very tight relationship with this normal coagulation pathway proteins and Alzheimer's disease incidence. So this is what happens in a clinical lab. How can we actually provide mechanistic studies in an aging mice model? What we did, we used intravital two-photon microscopy to assess the blood-brain barrier integrity in our HC57 Black 6 mice. For that, we had to equip the mice with a so-called chronic cranial window. We replaced the piece of a skull with a glass window, which provided a window into the underlying vascular function, which we were able to measure with two-photon microscopy by injecting different tracer molecules, different size of tracer molecules, and measure this fluorescent tracer extravasation in the brain parenchyma. What we did, we were able to create these cranial windows and measure baseline autofluorescence. Then we started injecting these dextrin tracers, which are normal compounds in the circulation, and measure how much of the different size of tracer is actually able to leave the vasculature and accumulate in the brain parenchyma, which indicated that in aging, indeed, there is a size-dependent extravasation of proteins in the brain parenchyma, which otherwise was supposed to be protected by the blood-brain barrier. And these extravasated proteins are able to trigger microglial activation and neuroinflammation. When I went to medical school, my understanding of blood-brain barrier disruption was that you inject Evans blue, and if the blood-brain barrier is not intact, Evans blue turns the entire brain blue. That's how medical students understand the blood-brain barrier also. But we don't really appreciate that doesn't happen at every single vessel level. It happens on the microvascular level. It happens at the capillary level. And it's a very focal reaction. So we were able to actually assess single capillary level blood-brain barrier disruption in young and aged vessels, showing that this is the site of the incidence where the age-related pathologies are starting, showing the importance of the microvasculature. Another structural phenomenon or phenotype that we find is the disappearance of these capillaries. These otherwise leaky capillaries or microvessels, for some reason, are disappearing in the aged tissues. This phenomenon is typical not only on the brain, but is also in the skeletal muscle, prevalent in the skeletal muscle, and in the heart as well. The disappearance of the small vessels contribute to decreased blood flow supply in the brain, decreased cerebral, overall cerebral blood perfusion. We were able to show both with two-photon microscopy and also with optical coherence tomography that indeed this is the case in our aged mice model as well. These pictures are an example how the young vasculature is intact, but the aged vasculature, especially at the microvascular level, is disappearing. The difference between two-photon microscopy and optical coherence tomography is that two-photon microscopy only reaches the cortical surface. So we showed that at the cortical level, we have less microvasculature. With optical coherence tomography, we also showed that we were able to image down to the hippocampus. So also the subcortical regions in the brains are suffering because of decreased cerebral perfusion. There is a new marker of aging, which is called microbleeds or microhemorrhages. And unfortunately, this has particular importance today. Cerebral microbleeds were recently discovered with increased frequency of T2 star-weighted MRIs. And originally it was so that this is just a side product, it's just a side symptom of hypertension. But now we understand there's these originally thought silent small bleeds that are less than 5 millimeters in humans. It's actually very active. They can progress, they can become multifocal, they can actually be the starting point for future stroke or larger bleeds. But what is even more interesting in that just the bleed itself can contribute to neuroinflammation and eventually cognitive impairment. These microbleeds are happening only in aging because of the vascular wall weakening. Previously, microbleeds were prevalent findings in Alzheimer's disease, primarily cortical microbleeds, and aging and hypertension. And that was primarily subcortical microbleeds. And the novelty is that now with the nondescript syndrome of long COVID, this is what we are finding in COVID survivors. The increase of microbleed number probably contributes to this nondescript neuronal symptoms that long COVID patients are complaining about. Nevertheless, in a collaboration with the neurology department and one of our good friend, Kaley Endowed, who was always interested in microbleeds, we launched a translational clinical study and we tried to find markers, easily measurable markers of microbleed incidence, beside of the otherwise very costly and time-consuming MRI images. And that we found that gait measurement, which every neurologist is using in the lab, can be actually a very good marker of higher cortical function. In our studies, gait stride length was one of the most sensitive markers of aging and age-related cognitive impairment. In recent collaborations with our biomedical engineering department, we started working together on a robotic device that actually it's a home-based robotic device that can track not only elderly, but also Alzheimer's patients. And based on gait parameters, probably indicate potentially decreasing cognitive function as well. This is all fine, but how can we model microbleed formation in mice? Well, mice do not develop spontaneous microbleeds. Mice do not develop spontaneous hypertension. So in the last couple of years, we came up with a mice model when we induce hypertension in a mice model with angiotensin injection in aged mice. And what is even more important, we think that the microbleed formation is not a part of a stable high blood pressure in the elderly, but more like induced by blood pressure fluctuation. The fluctuation of blood pressure is the one that is causing the pressure wave to burst the small microvessels in the brain. So we model this by the injection of angiotensin to increase stable high blood pressure, and then add name into the drinking water in a mice model to model the blood pressure fluctuation. And we were able to show that in mice, these microbleeds, which are less than 100 micrometer in diameter, so really small, this is not a major microhemorrhage, these are just small punctate bleeds, has a higher incidence. The neurological score or the neurological symptoms start much earlier in the age cohort, and the number of microbleeds are much higher also in the age cohort than in our young cohorts. What was surprising beside of this phenomenon is that we also found something that potentially can serve as a pharmacological intervention, and this is resveratrol. Resveratrol was always a very well-known vascular compound, which part of the French paradox that has beneficial vascular effects. And when I was a postdoctoral fellow here at the New York Medical College, resveratrol became famous, or so to speak, infamous, because of the aging research that originated from Harvard, showing that resveratrol has anti-aging capacity as well. So primarily we were interested in resveratrol because of the vascular function. And what we were able to show that resveratrol indeed, pretreatment with resveratrol indeed is able to prevent microbleed formation and protect the microvascular system in the brain, which we also validated with immunohistochemistry studies. as I mentioned before, gait was one of the best clinical parameters that we found as an easily measurable parameter for microbleed associated in microbleed formation. Now, how do you measure gait in a mice with 4 limbs? And whether we have a sensitive enough measurement to assess gait in mice. And here we used a so-called catwalk system. This is a fluorescent tube that the mice is able to walk, and each and every footprint is a paw print is recorded by a software. And the software is able to create to create a regulatory index for us, characterizing the footprint or paw print pattern in mice as an alternative for gait measurement in preclinical models. And we found that resveratrol treatment was able to significantly decrease the irregular measurement of gait or movement coordination. In recent years, my laboratory became more important, not just the phenomenon of microbleeds, but the underlying mechanism as well. So we think that microbleed formation is actually the pathological equivalent of the clinical transient ischemic attack. What happens is, and I hopefully can show, this is a laser-induced microbleed coming from the microvasculature, which induces astrocyte and microglia activation nearby. And if this microbleed is actually originates from the arterial, small arterial side, the pressure wave is so huge that it is inducing also vascular displacement. So this is an ongoing study when we try to measure whether the vascular displacement after this pressure wave of microbleed and the induced inflammation in the aged brain correlate with transient ischemic attack symptoms. I mentioned resveratrol as a potential vascular drug. My laboratory had several published studies when we used successfully resveratrol. However, resveratrol is notorious about its very low bioavailability. And we all know that resveratrol is really rich in red wine. But the amount of red wine that you should drink every day to actually reach a clinically significant dose of resveratrol is enormous. So not to risk liver cirrhosis, we have to have some kind of new delivery system for resveratrol and other potentially beneficial polyphenols. And my team started working together with a lipid chemistry group in Germany who patented a new liposome formulation called a physiogenic liposome. So the big difference between the regular or classical liposome formulation that's by now everywhere from cosmetics to drug formulations. The big difference is that the conventional liposomes are uptaken by the cells in the endocytotic manner, which also means that 70% of this liposome end up in the lysosomes and degraded without delivering efficient drug concentration in the cytoplasm. Compared to that, or physiogenic liposomes are fusing with the cytoplasmic membrane, and the cargo is directly delivered into the cytoplasm, several magnitude increasing the specificity over polyphenols, for example, as resveratrol is one of the best molecules to actually package by these physiogenic liposomes. So what my laboratory was able to show that in the brain, particularly, these physiogenic liposomes are accumulating and fusing with the vasculature, with the microvasculature. So these physiogenic liposomes are able directly to target the vasculature before any kind of neuronal damage happen, and also sometimes cross the blood-brain barrier. In further studies, we were also able to show that resveratrol delivered by the physiogenic liposome manner was significantly decreased the blood-brain barrier opening. So in our studies, we compared young and aged animals. Aged animals showed large extravasation of the fluorescent tracers that we injected in the mice. But administering resveratrol for 2 weeks, for example, with physiogenic liposome formulation significantly decreased this extravasation, this leakiness of the blood-brain barrier, literally closed the blood-brain barrier in aged mice models. So after these descriptive studies that we spent close to 7 years to put it together, I really became interested, what is the underlying mechanism? What is that biology of aging, cellular mechanism that can explain the vascular changes in the brain? And my hypothesis was that DNA damage-induced senescence is one of the common denominator that can link aging and accelerated aging-related cerebral vascular disease and cognitive impairment. So senescence says, or senescent theory of aging became kind of a fashionable theory of aging in the last couple of years, and the popular scientific literature calls these senescent cells zombie cells. Zombie cells because they are unable to die. They undergo damage, DNA damage. So these are damaged cells, but they are unable to die. And with aging, they are accumulating in tissues and organs. The word senescence comes from the Latin word senex, which means old age. But up until recent years, there was absolutely no scientific and research evidence whether this cellular phenomenon, this cellular DNA damage and DNA damage induced irreversible cell cycle arrest called senescence cause or contribute to aging. The hypothesis states that the steady accumulation of these damaged cells, senescent cells, actually has negative consequences on the body and contributes to aging. So what are these senescent cells? As I mentioned before, they are damaged cells, but they are unable to die. They are primed for programmed cellular death, but there is a step that actually blocks them to undergo programmed cell death or apoptosis. They are unable to fulfill their normal function, so their accumulation contributes to loss of normal cell function, which is basically the definition of aging. They are also unable to divide because these cells are arrested in the cell cycle at the G1, G0 state. So it decreases the regenerative capacity of our tissues and organs. And in recent years, it was discovered that these cells are highly secretory. They secrete inflammatory markers, cytokines and chemokines. And this links back to my immunology background, because this is how I started my studies, interested in these inflammatory markers. Now, the senescent cells are highly secretory, and the secreted inflammatory markers are causing a non-sterile, chronic, low-grade, sterile inflammation in the microenvironment, which is actually the other hallmark feature of whole body aging. Not only that, but also these secreted factors, which is called SASP, or senescence-associated secretory phenotype, can cause further senescence as a bystander effect in the nearby cells. Besides of cytokines, chemokines, these senescent cells also secrete several proteases, degrading enzymes that are contributing to the degradation of the nearby extracellular matrix, weakening tissue structure in aging. This was what was described for decades in a cell culture dish, but we didn't have any research evidence in vivo in any mice model, for example, or any other living in vivo model that this cellular phenomenon, cellular senescence, contributes to aging. Up until 2005 and 2006, when two groups independently, one at Mayo and one at the Buck Institute of Aging, came up with new mice models. And these new mice models were able to actually visualize the senescent cells, but not only visualize the senescent cells, but also specifically eliminate senescent cells from the body. And my laboratory was fortunate enough to get one of these models. This is the so-called P16-3MR model, a report on mice from the Buck Institute, Judy Campisi's laboratory. So I mentioned before that senescent cells are growth arrested. They are irreversibly growth arrested, and they are stuck in cell cycle at at the G0 step. So one of the molecules that is responsible for this is a cyclin-dependent kinase inhibitor called P16. And the mice model that we are using is using this fact. So P16 is activated upon DNA damage, and the activation of P16 in the senescence cells is paralleled by the activation or the translation of a luciferase enzyme to show in vivo senescence cell accumulation, also red fluorescence protein to be able to isolate these cells, for example, by flow sorting. But what is even more revolutionary, it had a herpes simplex virus thymidine kinase. This is the human herpes simplex virus thymidine kinase. And the FDA approved antiviral drug ganciclovir is actually act on this herpes simplex thymidine kinase, which means that by administration of ganciclovir, which is otherwise harmless in a mice, is able to eliminate selectively and specifically the senescent cells. So we potentially have a treatment option, an intervention option also to eliminate senescent cells. So in aged mice, this is what we actually confirmed. We were able to isolate senescent cells, especially senescent endothelial cells from the brain. We also showed with immunohistochemistry that senescent cells are primarily in the vasculature. The vasculature is very sensitive for all of the age-related noxious agents. Neurons are tough cells. Neurons are not the cells that are undergoing DNA damage. So we were able to show that senescence endothelial cell accumulation show an age-dependent characteristic. And further on, using novel technology, single-cell RNA sequencing, we were able to find markers of senescence. Cellular senescence is one of the cellular phenomenon that notoriously doesn't have any specific and unique markers beside of the cyclin-dependent kinase inhibitors. So by using single-cell RNA sequencing, we were able to create a new senescent cell score, a transcriptomic score, that now we can actually apply without the specific mice models, which is applicable also to human samples. So using single-cell RNA sequencing, we were able to identify different cell types in the brain. And we also showed that senescent cell accumulation primarily happens in endothelial cells and also in microglia cells. Further on, we were able to characterize endothelial heterogeneity in the cerebral microvascular system. So by using transcriptomic signals that was previously described specific, for example, venous endothelial cells or capillary endothelial cells or arterial endothelial cells, we were able to show that cellular senescence primarily happens at the in the capillary endothelial cells. This is something that's characteristic not to the macrovasculature, to the big vessels, but the small microvessels that Dr. Kaley and this department was so famous about investigating. This is all fine, but this is like a soup of information. How do we know where the senescent cells are localized? Do they show some kind of a predilection point or domain specificity in the brain, which can determine cognitive function outcomes? So I still remember the moment back in 2018, 10x Genomics just came out with a new method called spatial transcriptomics, which is able to combine single cell level transcriptomic study with spatial information. So what they do is they have slides, like normal histochemistry slides, pre-printed with 5,000 different array spots, and each array spot contains several millions of probes, uniquely identified probes. So by using this technology, what we can do is we can have normal tissue sections from the brain, but this is not histology. What you see here is not the histological slide, is actually based on the transcriptomic profile of each and every array spot, is the reconstruction of the different brain domains. And we were able to show by using senescence gene expression score that in aging, indeed, there is increased senescence spots expressing all of the senescence related genes. And this senescence cell accumulation compared to the young one is prevalent not only in the cortex, but also in the hippocampus and in the white matter as well. Now, this technology gives us a potential to see how this DNA damage pathway actually causes other gene expression changes in the same spots. So what is the consequences of a senescent cellular program? What we did, we compared the senescent cell gene expression with other gene expression pathways, and we did gene ontology studies where we identify different pathways in different domains, brain domain specific manner, together with the senescence course. And what we showed that, for example, in the isocortex, so the cortex, senescent cell accumulation or senescent cell gene expression is related to the regulation of adaptive immune response, immune system processes, and also with learning and memory. So this is the first proof in vivo that indeed senescent cells and the senescent cells secreted inflammatory factors are causing microinflammation in the cortex. In a similar fashion, we did these gene expression studies in the hippocampus, which is the primary site of learning and memory. And we showed that this senescent cell accumulation negatively affects synaptic function. It induces neuronal apoptosis and also regulates cytokine and inflammatory marker expression. We did the similar study in the white matter, and we found again immune regulation as the common denominator as an outcome from senescent cell accumulation and potential harmful effect in the nearby cells. Now, the beauty of this technology is not only that we were able to assess a transcriptomic change where senescence happens, but we are able to assess the transcriptomic change nearby of the senescent cells, which is the paracrine effect, how these released secretory factors affect the nearby cells. So what we did, we compared the senescent spots with the nearby spots. What is the gene expression change in the nearby spots? And the common regulator was again cellular immune response. So this is the first proof in vivo that indeed the accumulation of these damaged endothelial cells in the microvasculature cause neuroinflammation and negatively affect neuronal function as well. This is another study where we were able to compare microglia activation pathways in the same spot or nearby spots with senescent cell accumulation. What we showed that the resident immune cells, microglia, are the primary cells responsible for the chronic sterile inflammation in the brain. We were able to find and identify lymphocytes also, part of the adaptive immune response, but lymphocyte accumulation didn't overlap with senescent cell accumulation, which also shows that the immune response, the age-related decrease of the immune response, is partially responsible for the incapability to eliminate these senescent cells, which otherwise happens in a normal young tissue. So I started talking about eliminating senescent cells, this is really the potential for the next decade to develop novel anti-aging therapies. And the race is actually on to find new so-called senolytics that are specifically eliminate senescent cells from all the tissues and organs. And I just compiled here all of the biotech companies. Actually, this information is from 2020. So I assume by now the number is even higher. All of these biotech companies that right now are working to test novel senolytic compounds. So what are the senolytic compounds? You can eliminate senescent cells by using the regular immune response, which happens in young individuals, but the immune response in aging is also decreased. So if you can create some targeted immune cells that are specifically responsive to senescent cells, then you can rejuvenate the organ system. Also, you can actually counteract the effect of inflammatory markers. What we are using currently now is the fact that the senescent cells are primed to cell death. They are ready to die, but there is a link, there is a step that inhibits them. So the current senolytic drugs are pushing senescent cells toward apoptosis. And one of them is the FDA-approved anti-cancer drug, navitoclax, ABT263, that has very potent senolytic effect. So in more mice models, both in the P16 mice model, as I mentioned before, ganciclovir, an antiviral drug, is able to eliminate senescent cells in that particular mice model. We compared that one to just regular C57 black 6 mice treated with ABT. And we showed that we were able to selectively eliminate senescent cells from the brain. We also showed that elimination of these senescent cells has very beneficial effect on the neurovascular coupling response. So the cerebral blood flow increase upon neuronal activation, both using our marker mice model, but also using just a C57 black 6 aged model. We also showed that elimination of senescent cells actually prevents the blood-brain barrier integrity loss. In our studies, we measured the blood-brain barrier integrity in aged mice. Then we started with a ganciclovir treatment regime. And several months later, we measured the blood-brain barrier leakiness. And we showed that after 1 month of ganciclovir treatment, the leakiness of the blood-brain barrier was decreased. And it was further accelerated after 3 months, which would be wonderful. But the situation is not so simple, unfortunately, because we followed these mice further on, not just 1 month and 3 months post-treatment, but also 6 months post-treatment. These are really old mice by this point. So is this effect of the senolytics is sustained, or do we have to repeat all the time the senolytic treatment? And what we showed actually was that the peak of the effect, the senolytic effect, we were able to measure after 3 months. But after 6 months, the senescent cell accumulation took over again. So meaning that most certainly in any kind of new interventional design, we need to keep up the senolytic drug administration. We also showed that eliminating the senescent cells is beneficial to revascularize the brain tissue in a safe manner. And the ultimate proof is whether all of this vascular improvement after senolytic treatment has a beneficial effect on cognition, because after all, this is what we want to target in our human studies. So what we did again, radiogram water maze testing of my spatial learning and memory function. And although we were able to show that new information, the learning process was not improved in these mice, which are very old. They are equivalent to 80 years old humans. But new information processing was actually improved. So this study is forcing us to go back again to the laboratory, start all over again, to identify that window of treatment when we can successfully not only rejuvenate the vasculature, but still have an effect on neuronal function as well. So as a summary, aging-induced activation of the senescence program has negative effect on the microvascular function and structure, decreasing the vasodilator function of the aged vasculature, and also opening up the blood-brain barrier, contributing to neuroinflammation and neuronal deficit. But elimination of senescent cells has beneficial effect in terms of rejuvenating the vasculature and hopefully also the cognitive function. So conclusion, cerebrovascular senescence is a focal phenomenon creating points of least resistance in the microvasculature. The chronic accumulation of the senescent cells is actually overlaps with all of the phenotypic and functional changes in the aging brain. And cerebrovascular senescence contribute to uncontrolled low-grade chronic activation of innate immune system in the brain. So where we are going from here? So the ongoing studies try to identify what is the trigger for vascular senescence. What is the factor that actually causing the vascular cells, endothelial cells to undergo DNA damage program? in our new studies, we try to investigate, for example, circulating factors with heterochronic parabiosis models. We also want to know whether elimination of senescence cell attenuate eventually neuroinflammation, whether we can reverse microglia activation in the brain, or untangle the cellular network in the vicinity of the senescence vascular cells. And in the future, maybe design new, safe, targeted delivery of senolytic compounds. Currently, the mostly used senolytics are anti-cancer drugs. We don't want to use anti-cancer drugs with serious side effects as an anti-aging compound. So whether we can deliver this compound selectively to the brain is a big question. And we also want to explore new other strategies to eliminate senescence cells, maybe with targeted immune therapy. So last but not least, this is my very talented team, graduate student in the first row in Oklahoma, but also all of those young faculty members who actually selected me to work with me. And this is a very multidisciplinary team, which has bioengineers, medical engineers, public health experts, MD, PhDs, veterinarian medicines, and mathematicians working together to untangle these problems. And this is the life lessons that I learned from Dr. Kaley, how to build a team. So be always strong and very supportive. This is what he gave to me. He believed in me, despite of my tarnished pedigree in terms of vascular physiology, but hopefully I was able to convince you that I made up for that. He created a very secure environment. My postdoctoral years were the happiest for me. I didn't have to be worried about budget or financing because he created that safe background for us to be able to focus, critically focus on successful science. He always conveyed his belief in the success of his students and their abilities. And he always wanted us to go and be multidisciplinary. This is how I got here as an immunology student. And last but not least, food and cuisine can always bridge cultural differences. I have never seen him happier than at the departmental party, and he was able to offer his own homemade goulash, a Hungarian food, and I was so proud to be a fellow Hungarian. Thank you so much for your attention.
We'll enter the Q&A session, and I have a mic that's going to go around, but Dr. Wolin has a question first, so he gets to go.
Anna, thank you for this fascinating talk. One thing you mentioned early on at the beginning was that presumably removing hydrogen peroxide by mitochondrial hydrogen peroxide by increasing catalase had protective effects. And you didn't elaborate too much, but I'm kind of wondering what was that beginning to be driving, and did it end up being something that goes on to senescence if that was the way your talk ended? So it would be interesting. Signaling mechanisms, processes turned on, whatever comes to mind.
So what we know that overexpression of hydrogen catalase, which eliminates hydrogen peroxide from the mitochondria, has protective effect in terms of cardiomyocyte function, endothelial function, and vascular smooth muscle cell function. And the underlying mechanism, what we investigated from an endothelial perspective, is actually the increased free radical production, which is able to scavenge NO and 4-peroxynitrite, which contributes to the decrease of age-related NO depletion. Whether that one has any relation with senescence, right now we don't know.
Thank you.
Dr. Fisher's up next.
That was a really amazing talk, particularly all of the imaging modalities they used. It was really, really cool.
Thank you.
So I have a question about the neurovascular coupling measurements that you did. In the aged animals, do you think the impairment, what do you think best explains the decrease in neurovascular coupling? What aspect of the So it sounds like it's mostly vascular effects, and what aspect of the vascular effects?
So I used to get the question, the chicken and egg question, how do I know that vascular changes precede neuronal changes? How do I know that not neuronal dysfunction, age-related neural dysfunction drives the decreased neurovascular coupling phenomenon? Is this the question partially? Okay, so what we did, we measured evoked field potential in these young and aged mice. So basically, we are triggering the neuronal function exogenously, and neurons are firing fine. So neuronal function by itself in aging at this point is not decreased. So this is how we concluded the vascular loss of integrity, functional and structural integrity, precedes the neuronal dysfunction. So what can be the underlying mechanism in the neurovascular dysfunction? We try to detangle that complicated slide that I showed in terms of different signaling mechanisms. And what we found that EETs, which are potent vasodilators, are significantly decreased in aging. And 20-HT is significantly increased, which is a potent vasoconstrictor. So that's one of the underlying mechanisms. And Anne, as an EET expert potentially, can comment on even more further on this one. But the common denominator is the decrease of NO. And we did these studies not only intravital, in an intravital preparation, so in live animals, but we tried to do it in hippocampal slices also, or cortical slices. And it was always coming back to NO. And I know that when I came here, NO just became, it's like fashionable molecule and fashionable target. But this is science. So we are going back again and again with 3-4 different methods. And we always end up on the common denominator is NO.
We have Dr. Gologoski next.
Anna, thank you so much. Your talk spans on several levels, from molecular to the whole animal, mouse, and then to human. It was, if I may say so, kaleidoscopically spectacular.
Thank you.
Anna, the question that I have is, based on the analogy with, say, glomerular capillaries, people observed that there are in senescent or in hypertensive animals, there is a formation of microhernias in the capillaries. I wonder whether similar microhernias have been seen in brain capillaries. I think in one of your slides, I thought that those might be microhernias, and whether they might represent the sites of future microbleedings.
I wish with all my heart that I could answer this question. And it's actually a very valid point. When we started these studies, I never realized that the vasculature is such a dynamic organ in itself. So neuroscientists are able to go back and record the same neurons again and again and again live. We are not able to do that, because if you go back to the same spot 2 days later in an intravital in vivo mice preparation, you won't be able to see the same picture. The vascular is changing so much, so quickly, that finding exactly the same capillary is almost impossible. Just locating the same microbleed is very tedious. So I truly hope that maybe with a new and more specific microscope that I hope to get, the Leica Stellaris microscope, by the end of this year, we're gonna have a much better temporal resolution and be able to go back and maybe address this question. But as of now, unfortunately, technically, at least my team is not able to do that, not in the brain.
Anna, thank you very much for a very exciting talk. as a point of clarification, your mouse model, do they develop hypertension as they age?
No, no, we have to induce hypertension
Okay.
by osmotic pump infusion of angiotensin. No, C57 black 6 doesn't develop spontaneous hypertension.
Yeah, the reason why I asked that question was because I was attempting in my own mind to dissociate the aging process from the effect of hypertension, given the work of Phil Hutchins in the 70s and 80s, where in young animals that experienced a hypertension either spontaneously or hypertension induced, there was a rarefication of the microvasculature.
Absolutely, absolutely. Hypertension, and we have studies that showed that hypertension in aged mice are potentiates the vascular rarefaction phenotype. So hypertension contributes to the disappearance of the microvasculature.
Yeah. So then in the absence of the hypertension, there's no cortical thinning, which would occur, and there's no direct effect of pressure on the endothelial cell. So this is then, in fact, a true effect of aging.
It's a little bit more complicated and more controversial now with the cortical thinning part, because what we understand now that in healthy aging, the number of neurons are not changing. In healthy aging, the neuron number is not decreased. Neuron number is only decreased in neurodegenerative diseases like Alzheimer's. But in healthy aging, that's not the primary problem. Synaptical plasticity or synaptical dysfunction happens much earlier. But losing the cortex or cortical thinning or losing the number of neurons is primarily associated with neurodegenerative diseases and not aging per se. And that brings up the problem also, how can you differentiate between aging, healthy aging, health span, and age-related diseases?
Thank you.
We have a few questions online. Akos wants you to comment on, can exercise be an anti-senescence agent?
I don't know, but exercise is definitely one of the best intervention to induce angiogenesis in the brain in a safe, VEGF-independent manner. What we know is there is vascular rarefaction in aging, not only in the brain, but also in the heart and the skeletal muscle. And that vascular rarefaction is not responsive to VEGF treatment, vascular endothelial growth factor, which is a potent vasculogenesis, angiogenesis inducer. Right the contrary, in aging, the circulating VEGF level is super high. And that's also from our own translational clinical studies. The endothelial cells are irresponsive to the VEGF-induced angiogenesis pathway. So exercise is the one which in a non-canonical VEGF-independent manner is able to induce vasculogenesis, angiogenesis in aging. So the beneficial effects is definitely there at the molecular and cellular level as well.
Thank you.
I hope that it's.
Next one is from Dr. Zucker. Can you comment on the differences between males and females on the blood-brain barrier and behavioral changes with aging?
Yes, we measured both male and females, and these are female mice in menopause, and we didn't really see any differences in terms of loss of blood-brain barrier integrity, or at this point, behavioral characteristics. So it seems to be a universal phenomenon. Although I know that, especially with the angiotensin-induced hypertension studies, that I'm aware of those studies that show sex-dependent phenotype.
Is Edwin Namato online? Do you want to ask your question?
Yes, thank you very much, Dr. Csiszar. I have a question. Do you think that the vascular senescence changes that you demonstrated could be attributed to the effects of low flow in capillaries and shear rate that has a detrimental effect on endothelial function and indeed inflammation through the glycocalyx?
I don't know. I have to be very honest. I don't know. This question is a really hot topic now in the vascular field, how we can actually study the glycocalyx. What we know that the glycocalyx, which is outside of the in the luminal surface of the endothelial cells, undergo a very dynamic and very fast change based on the environment. There are devices that are able to measure glycocalyx changes in human oral mucosa, and just by having lunch actually triggers a huge glycocalyx thickness change. So we contemplated this idea several times, how to address this dynamically changing part of the endothelium, especially with our liposomal studies, because we know that the glycocalyx serves as a net, getting all of these liposomes in the microcirculation and directing the liposomes to endothelial cells. Right now, I don't really know methodologically how to address these questions in vivo, because I think that the in vitro models are really lacking in terms of recapitulating this dynamic change of the glycocalyx as a response to the environment. But this is something that it's actually a hot right now.
That was an excellent talk.
Thank you.
How does this all tie into
Welcome.
and senile amyloid itself? These vascular changes.
I apologize. Would you be so kind to repeat the question?
The association between these microvascular changes and the development of senile amyloid.
Yes. I'm not particularly an Alzheimer's scientist or researchers, and I always promise myself that if there is a topic that I won't ever to touch is Alzheimer, because of the amyloid plaque and the tau currently prevailing theories. Because what I think is that Alzheimer also has a very, very strong vascular component. If you see all of those earlier pictures from the early 2000s, all those amyloid plaques are actually sitting on the microvessels in the brain, literally constricting the microvessels. And we know that the clearance function of the vasculature is decreased in aging. Unfortunately, the current mice models that we have to investigate Alzheimer's disease are all based on the early onset Alzheimer's disease, the familiar Alzheimer's disease, which is only 5% of the whole Alzheimer's disease population, and doesn't address the age-related late onset Alzheimer's pathology, which I think very much shows an interplay or an interaction with the microvascular system. But I think that the current models are not able to address this age-related phenomenon.
Yeah, my question is actually a little bit related to the last question from Dr. Frischman. There were some studies over the last several years that showed that beta amyloid could bind to parasites in the cerebral vasculature, particularly in capillaries, and trigger contraction as an explanation of a reduction in the cerebral blood flow. So actually, my question was also whether or not there's any evidence of the parasites as a target and involvement in any of the things you were talking about, you know, as an intermediary in maybe involving beta amyloid, but also whether that the parasite being affected can open up the blood-brain barrier.
Absolutely. Thank you so much for this question. My group doesn't work on this topic, but I showed one of the slides from Berislav Zlokovich's group, and this is what he is investigating. His hypothesis is that in Alzheimer, the first step in the disease cascade is actually hypoxia-induced pericyte damage in the microvasculature, which causing the opening of the blood-brain barrier, extravasation of the normally circulating coagulation cascade proteins like thrombin and fibrin, which also can serve as a focal point for amyloid aggregation. That's an excellent question. Yes.
Hi, so thank you. I just want to say as an MD PhD student, it's very inspiring to see the complexity and the synergy of all the work that you've done that's so clearly motivated by the translation. I was interested in the delivery system that you mentioned earlier with the fusion lipid.
Fusogenic liposomes.
Fusogenic liposomes, yes. So when it's not fused and you get the lysosomes removing things, would you be worried about introducing synthesized lipids into the cellular membrane and disrupting, any of that, or is the amount so little that it would be
Yes,
negligible?
absolutely. We did these studies, and this is actually very difficult to address in vivo. However, what we have seen and what we know that the aging lipid bilayer loses its flexibility. So it becomes rigid, and actually introducing these fusogenic liposomes improves the flexibility, the motility of the lipid bilayers. So not only we are able to target the cargo directly in the cytoplasm, but we are rejuvenating the cellular membrane as well. You're welcome.
Anna, thank you very much for really a wonderful talk. It's perfectly appropriate for the occasion as well.
Thank you so much.
Indeed. I have a question though about the spatial dimensions that you talked about a little bit. I think with diseases like senile dementia, various dementias, there's often a localization or vulnerability of certain regions in the nervous system that are greater than others. Does any of the work that you've done on the vascular bed suggest that vulnerability comes about from the circulatory system, or is that vulnerability preferentially on the neuronal side?
This is what we try to detangle, and I mentioned this evoked field potential measurements that try to assess the neuronal health, at least in the cortical regions. Now, the spatial transcriptomic studies are already, it's a cross-sectional study from a certain point, so it's not dynamic. We don't know what's going on with the neurons at that point. What the behavioral studies implicates, though, that if our hypothesis is true, the whole pathway starts from the vascular side, but we eventually lead a critical threshold level of neuroinflammation that affects neurons, that affects the synaptic density, synaptic plasticity, and eventually cause irreversible neuronal damage. And that's what our hypothesis is, why our senolytic treatment at such a late age wasn't effective on cognitive function, improving cognitive function, only vascular function, because we are too late. So we think that it's a cascade. It's a very slow and gradual cascade, but this is our understanding right now, or this is our hypothesis, or a comprehensive framework how we can explain it.
But then you would imagine that vascular bed, certain parts of the brain might be more vulnerable.
Exactly. And that's why that's why we did all of those spatial transcriptomic studies to see whether the transcriptomic profile is different in the cortex or in the white matter, in the hippocampus. And yes, it is different, but the common pathway that it's triggered everywhere is inflammation and an immune response. And I have a couple of other studies, ongoing studies, other projects with accelerated aging. For example, in chemotherapy-induced vascular senescence, or radiation therapy-induced vascular senescence. We wanted to see whether in those cases, which are non-triggers for DNA damage and cellular cell accumulation, in those cases, we can see a similar accumulation of senescent cells, similar brain domain specific accumulation. And what we found is, although the phenotype is similar, there is cognitive impairment, there is spatial, decreased spatial learning and memory function, there is vascular senescence, but the vascular senescence accumulation shows a completely different picture. It's primarily cortical, for example, in chemobrain, which is just one of the biggest problems in cancer survivors, that they are losing their cognitive plasticity.
Thank you so much. We're going to end the question period and we're going to move on in the program.
Thank you, Anna, for really such a wonderful lecture today. It's so much also built on what you started with Gabe and in endothelial dysfunction with aging. It brings great honor to him today as well. And I have the pleasure of actually awarding you the plaque for today's lecture.
Thank you so much. Thank you. It was truly an honor to be here today.
Thank you for such a wonderful lecture that you gave us.
And we will have closing remarks by Dr. Messina. Here is Dr. Messina.
Good afternoon, everyone. Is this loud enough? Maybe I should speak louder. Okay. Welcome, everyone. There are a couple of things I'd like to say before I discuss a few items about Gabe. First of all, I should like to thank once again Harriette and David Kaley for sponsoring this legacy, this memorial legacy for Gabe. It's a very special event in my life and in Irv Zucker's life. With both the bookends here this particular day, because Irv was Gabe's first student and I was his second student to finish PhDs under his mentorship. In addition, I'd like to thank Marcello and Michael for their contribution to allowing this lecture to go forth the way it has for the last 9 years. In the early days, I was the one that ushered it around. And then since I retired, it's been in the good hands of Michael Wolin, and now more recently with Marcello as a partner. I also want to thank Anna for several reasons. One, because you have validated one of the Messinaisms. Those of you that had the privilege or the honor or the misfortune to come to one of my lectures for either graduate students or medical students, I was known for the so-called pearls of wisdom that were identified as Messinaisms. And I thank you for validating one of my Messinaisms, namely that if you live long enough, you become a victim of life. So I thank you for that, because now you have scared the hell out of me. Because in addition, I've learned from you that if you live long enough, you become your own risk factor. And given my longevity, I now realize that I'm a living risk factor. So I want to thank you for that. But in seriousness, this was a wonderful lecture, and it is a tribute to Gabe's legacy. And we thank you very much for that. Something about Gabe. Well, in Harriette's earlier remarks, she said that Gabe wasn't scarred, or reported that he wasn't scarred by his experience in the concentration camps, but was only marked by it. But I want to think back about that, because Gabe's sense of humanity and family really was the hallmark of him as a human being. His approach to mentoring, his approach to managing the department were entirely family-oriented and food-oriented. And so his dreaming of a menu that would sustain him after his escape from the concentration camp, which you should all know, he did escape eventually with his partner, Tom Vaghi, as I recall, giving the stories that I heard quite frequently. He always dreamt about food. And so you heard about the history of the Department of Physiology having lunch together as a group, which fostered a lot of interaction and a lot of cross-fertilization for very productive research. In fact, that research stands as a legacy here in New York Medical College, alongside of those that also established legacies here. the likes of Ward Pigman in the Department of Biochemistry for his research on carbohydrates and the discovery of the only soluble carbohydrate, namely starch. I don't know whether or not anybody's discovered another one. And the likes of the chairman of Anatomy and Physiology, who was an outstanding electron microscopist along the likes of George Pilati. And of course, somewhat more recently, that of David Lair, whose contribution to the research of sulfur drugs when antibiotics weren't important here. These gentlemen have left a legacy of outstanding science. Gabe is among them, and I'm proud to have been his second student to graduate with a PhD degree. But that food theme in his life also extended to when we as students, Irv and I, would travel with Gabe to the various scientific meetings. Gabe always insisted that every student that was making a presentation at a scientific meeting would have the pleasure of joining him in what was known as the dinner. We came to know it as a time to eat and to provide sustenance for ourselves. Those of you that were in Gabe's department always knew that there was a meal waiting for you at the end of a scientific meeting. One of the things that also was outstanding about Gabe is his innate understanding, just as that quote by St. Georgie was provided by Gabe, by Irv today as a reminder that it is important to be able to see beyond what others see when something is discovered. And so I gave Gabe the nickname of the nose. He wasn't too happy about that because he always thought it was because of his nose. It was never his nose. It was his natural instinct in terms of where to go scientifically. And I'm reminded of what happened, and I happen to be present so I can document this, when a salesman from Upjohn walked into his office. Gabe and I were discussing some experiments. And this gentleman from Upjohn said he had some new compounds that he would like tested. And Gabe said, sure, why not? He was always open-minded about new explorations. That was the other thing that excited Gabe, exploration, going where no one else had gone before, and the discovery component of it. And so he took these compounds with Richard Weiner, and the two of them discovered that these compounds were pro-inflammatory and chemotaxic. And then a little Italian guy by the name of Messina showed up and demonstrated that these agents had some effects on the microcirculation. Well, those three discoveries, two of which Gabe and Richard Weiner conducted together and one that I contributed to, provided for the hypothesis to Salvatore Moncada and John Vane that there was a commonality amongst these three elements that suggested that these compounds that were newly discovered and provided by Upjohn were pro-inflammatory, and therefore the agent that was best known to prevent inflammation, namely aspirin, probably was acting by way of inhibiting prostaglandin production. And that's what those compounds were known as, prostaglandins. In fact, John Vane and Salvatore Moncada, John Vane having gotten the Nobel Prize for the discovery of the action of aspirin, were frequent visitors to Gabe's lab and always credited him with the findings led to their hypothesis and for them then getting the award. So Gabe has contributed not only here to the New York Medical College, not only to individuals that were privileged to be in his presence as either students and then later as colleagues, but also to humanity as well on the large scale because of the discovery of the actions of aspirin, which were directly dependent upon prostaglandins for their inhibition. So I want to thank everyone for attending. And I can't believe that 9 years has gone by and 4 years since I have retired. It's just amazing how time goes on. And I also want to thank you for one last thing. That is, given your work, I now realize I have to increase my wine consumption. So with that, I thank you.